Hi, my name is Amir Tataibi and I'm your host at Intelligent Medical Decision Making Seminar Series. Today we have Dr. Farzad Farahani from Johns Hopkins University talking about daily variations of brain connectivity patterns, a graph-based analysis. Dr. Farahani is a postdoctoral researcher in the Department of Biostatistics at Johns Hopkins University since fall 2020. He earned his PhD in the computational neuroergonomics track of the Industrial and Systems Engineering program at the University of Central Florida in 2020. Thank you very much for being with us.
Natural, biological, internal 24-hour, approximately 24 hours of fluctuation that have been observed in many animals, most of, better to say, almost all of organism, including animal, plants, fungi, and even bacteria. So it is not only for human; it's almost for every organism. And this 24-hour fluctuation regarding the human performance is like this, is going to be increased during the day and decreased at night. And for example, here at the bottom of the figure, midnight, when we go to our morning, 4 a.m. That we have the lowest around 4 a.m. We have the lowest body temperature, and then afterward we have, for example, at 10 a.m. we have the high alertness of the body, and then during the afternoon around 3 p.m. 3:30 we have the fastest reaction of the body in terms of the coordinate between the body and the brain. And for example, at 5 p.m. we have the best coordinate between muscular activity. And so many scientists, many researchers believe that it's the best time for doing exercise that need more muscular activity. And then go on for example, 9 p.m. or 21, the melatonin secretion starts, and this is the hormone that makes us feel drowsy and sleepy. And for my study, I wanna see what's the difference between the morning. By morning, I mean 1 hour after waking up the participants, and evening. We have 2 sessions. We want to see what are how network properties and connectivity pattern will change between these 2 sessions. And the evening is 10 hours after waking up. Around, for example, if someone wake up at 10, and then 10 hours later, or someone wake up at 7, it depends on the chronotype of the participant. So as an average, for example, we can imagine what's the difference between 8 or 9 and 6 p.m. evening and morning sessions. We can go to the next slide. There's a nice fact in this way, relevant to the circadian rhythm. It's about the pineal gland. It's a gland within the brain. Some believe and say to the pineal gland as a third eye in the human brain. The reason why it's called it, because when lights reach the eye, the photoreceptors behind in the retina will receive the signals, and then transmit the signal to the hypothalamus, the red small circle in the middle, transmit to the hypothalamus, and then the hypothalamus will transmit the signal to the pineal gland, and the pineal gland secrete melatonin that make us drowsy and sleepy. For many years, people was thinking that it's an extra organ in the human body, and it's not. There's no benefit, and it's not useful. But now, the pineal gland is the single only manufacturer of the melatonin, and the one of the most important factor in this circadian rhythm. You can go to the next slide. Thank you. And here, I want to talk the effects of circadian disruption. What happens if this circadian rhythm is disrupted? There are many studies and research that show the correlation between the disruption in circadian and many disorders such as obesity, diabetes, and also some neurodegenerative disorders such as Alzheimer and Parkinson disease that are mostly studied in the literature. We can go to the next slide. So far I talked about circadian rhythm, and now it's time to talk what data we want to use. We have two sessions, evening and morning sessions, and we want to scan and access the fMRI or functional magnetic resonance imaging from participant at these two sessions. What is fMRI? In the top left, you can see the MRI scanner. It does have some major components, a big magnet that creates the magnetic field for us. There's gradient codes that cause the magnetic field to be rotated and get the best possible resolution of the image when the patient is lying on the scanner. And also there is a radio frequency coil that transmit the radio frequency signal and also then receive the projection of the signals after hitting to the human body brain. And the way that the fMRI is working is by detecting the changes in blood oxygenation level. So if a brain area is active, it needs more oxygen, and then fMRI is a way to get these change of oxygenation level in the brain. There are some other similar imaging method that work like based on the difference of some other things, but fMRI is based on the difference that the machine grab based on the oxygenation level. For example, in the left button, you can see those active area that are red, and also in the right button, you can see at rest. For example, there is less oxygen in the blood vessels, and in the right one we can see more oxygen that are tag along to the hemoglobin. So just here to show what is fMRI and how it can work in simple words. And we can go to the next slide. fMRI are considered as time series. How we consider them as time series? As I said, fMRI will trying to get the amount of neural activity or the oxygenation level from the brain. So if we scan a subject across time, we can get like a time series that in each point we have one time point that shows the fluctuation or neural activity of that participant. And here we can parcellate or divide the whole brain as several lots of cube said voxel. For each voxel, we can extract and get the time series for that specific voxel and analyze that specific one or its connection to other voxels. So just want to say that fMRI is a sort of time series, that any kind of time series analysis can be applied on fMRI data. We can go to the next slide. This is the general flowchart and pipeline analysis for the study today I want to talk about today. We have fMRI time series from different participants. Then after some pre-processing that is very important in any kind of imaging analysis. After some pre-processing step like aligning the data based on different subjects and registering coordination, we can go to the parcellation step, the C, to parcellate because each brain approximately, just as a number, it does have around 90,000 voxels, and it's extremely difficult and complicated to analyze all these voxels at this resolution. So we need to parcellate. Then in parcellation, we can increase the signal to noise ratio as well, because all subjects are not going to be aligned the same way. There are some differences in the brains and the structure. So this parcellation can help us also increase the signal to noise ratio. And then after parcellating and dividing the whole brain to these amount of regions, as you can see in the C panel in this figure, we can extract for each region a time series. This is the average of time series for that region across all voxels. And then based on these time series that you can see in the D panel, we can create a correlation matrix that is a functional connectivity matrix showing the statistical dependency between different regions. So this matrix is a symmetric square matrix that shows that the correlation of the pairs of regions inside the brain. And the next step is optional, but people usually do to both make it less complicated and also get the most important information from the brain, and then binarize the correlation matrix by setting some threshold. For example, just considering those correlations that are higher than a threshold, those strongest connections, those strongest links in the brain. And then based on this binarized connectivity that you can see in F panel, we can create the functional brain network as shown in G. This is a network that shows the connections between nodes and different regions of the brain by some set of links that are connected. And then using this functional brain network, we can do network analysis and extract some graph theory-based measures to compare the groups that we have. For example, here in this study, we have two groups that are corresponding to two sessions, morning and evening. We can go to the next slide. Thank you, Dr. Tavi. This is also, again, to show those functional connectivity. You can see the top two figures are the correlation matrices for evening and morning, and the bottom two are the binarized one after thresholding and binarizing the correlation matrix. Here you can see that inside the axis you can see some rectangles that shows different systems inside the brain. Systems can contain different regions. For example, a set of regions can be aggregated to each other to form a system. The purple one, as you can see, is the visual system. The blue one is the motor system, and so on. We have for left and right hemisphere as well. I just plot this figure to show you how these connections exist between different regions of the brain based on the extracted time series for each region. We can go to the next slide. And anytime, because of this this issue that happened, and anytime if you have any question, feel free to ask. And yeah, that would be great to stop making any confusion. After creating the functional connectivity and those correlation matrices, it's time to introduce some measures or features based on graph theory. In this figure, you can see a set of global measures. Global measures are one measure that representing the brain as a whole. For example, the average shortest path is a measure. What's the shortest path is the path that connect two nodes with the minimum possible of connections. So if we average all possible connection between all pairs of node in the brain, we can get a feature as named shortest path in B. You can see in B, and it can be a measure representing the brain. And for example, in this study, we could compare this shortest path between the evening and morning session to see if the path, the information flow, what does it mean? This path is correlated with the information flow inside the brain, how information is integrated, how efficient is the flow of information within the brain. Or other measure like clustering coefficient is a measure that calculates how the neighbors of a given node, for example, in this figure, the middle one is the node, yeah, exactly. And we want to see how interconnected are the neighbors of this specific node. This is another measure just to show the clusters existed cluster in the brain, or communities, for detecting communities. And there are other measures like, as you can see in D, assortativity. It is a measure that calculates the resilience of a network to failure or dropping out a specific node. How resilient is a network if by random we remove one of the node? It will collapse or is robust against this failure. And there are other measures. And in next slide, I plotted some results based on the global measures. As you can see, the sparsity, the axis on the sparsity, is just about the binarization. By 5%, I mean 5% of the most important connection. Here, for example, I just considered 5% of the most important connections, and so on. Just assuming the first sparsity, comparing morning and evening, morning shown by yellow and evening by blue, you can see the small wordness has increased. This small wordness or sortativity and other global measures together are showing how efficient could be the brain between these two sessions, when we compare these two sessions together. And as a general, we can see at this 2 time of day, the morning 1 hour after waking time and 10 hour after waking time, we have seen some increase in efficiency of the network based on global measures. These are just global measures. They could be there are some challenges in network science that these measures are not that much informative. For example, what does it mean that the assortativity of a network is bigger than that one? But as general rule of thumb, they are about the efficiency of networks, and uh, here we can see the efficiency has increased after some hours. The brain is more efficient, is more integrated, and more segregated, and after some hours of waking time. We can go to the next slide.
And since we have a question here, I think there was a question here. They asked, how do you define efficiency?
Yeah, this efficiency again, if we, for example, the shortest pass, if we consider the shortest pass as a measure, information flow, or the amount of connections that need to be passed by a node to reach another node is decreased. I mean, we have, for example, we have less number of connections to be passed to connect two nodes, or a pair of nodes, or pairs of nodes within the brain. So we need the information flow is more integrated. We have uh, we have we have better information flow considering less wiring cost. It's like an optimization. We want to make a balance between the wiring cost. We need less connections, less wiring cost, but we need to connect nodes as much as we can to make it the whole network integrated, more integrated. Just by efficiency, I'm talking about this. The efficiency is the opposite of shortest path. If we have, for example, longer path, the efficiency is less, or the opposite. Increased, another question I got, increased sparsity implies better synchron. No, it doesn't mean that implies better synchronism, because sparsity is just about the number of connections that we use for creating the network. And it means that we considered most of nodes and connections. We considered the network in more general form than just those strongest connections. And this is about the density of the network. Sparsity here, by sparsity I mean only the density of the network. For example, by 50% means that 50% of the nodes and connections are considered. And by 5%, we lost many of those connections and nodes. Another question we have: 50% of the nodes or the edge. These are about nodes, 50% of the nodes. But in relation to nodes, we will lose many of the edge. But this sparsity is defined based on nodes, number of nodes. Okay, cool. We can go to, yeah. And after global properties, we can define some local properties. Local properties are those that are defined for each node. For example, the degree of a given node. These properties could show us the centrality of a node, how important is a node. In this way also we can detect the hubs, the most connected nodes in the network, and compare the centrality of nodes again in these two sessions between morning and evening. And it's a good way of analyzing the whole network just by focusing on each node separately. And this here, it's not again like it's not like global that each measure is representative of the whole brain. Here each measure is for each node, and we can compare each node in the same node in both sessions. And we can go to the next slide just to show some results. For example, in this study, in this table, you can find some most informative and most significant changes between morning and evening. By the first column is the region of interest number. Second, third, and fourth is about the MNI coordinates or coordinates of the brain of those regions. And next column is the name of those regions. And then we can see the p-value for 4 different measures, local measures that mostly they are correlated. Just assuming in the first one, degree centrality, we can the values that are in the table are the p-value, the significant p-values that are detected between the morning and evening sessions. And in the next slide, yeah, here for example, again the same table. I specified some regions by a rectangular a line, red line, you can see, and these areas are, based on the literature, are related to language system. And it's interesting that to see that, for example, the language system, how the language system of the brain works will be affected after passing some time during the day. The next slide is about the sensory motor, the motor cortex, and the other parts. Again, I specified them by this red rectangle, and you can see that how the brain could be affected during the day, how the motor system and the motor activity of the brain could be affected after passing some times across the day. And the next slide is about the limbic system again. It's interesting. These are 3 important systems or network that during the study, during my analysis, I found that could be affected more. The language part, the language processing system, the motor control system, and the limbic part that is responsible for emotional and behavioral activities inside the brain. And yeah, these local properties are, in my mind, are more informative because you can find exactly which region, what exact region could be affected more in terms of the conditions or sessions that we have in the study. These sessions could be any session. For example, someone can work on neurodegenerative disorders or like Alzheimer's disease, and compare 2 groups of healthy control or Alzheimer's disease together to see how these properties, network properties, could be affected, or any other definition of the sessions or groups that we could have based on the data. These are applications that the graph theory could be helpful and shows us how the network properties of the brain could be changed in terms of global and local properties. And if we have next slide, yeah, here also this is the plot just to show In the x-axis, we have those regions. Assume that we have 200 regions based on the atlas that I use, and in each region, I showed both for the morning and evening. Morning in every figure that is shown by yellow, and evening is shown by blue. And the red dashed line red dashed lines show the significant changes across these regions, and each panel is representative of a measure. For example, A is regarding the degree centrality, B betweenness centrality, and the C is nodal clustering coefficient. These are all local properties. As I said, many of them are correlated to each other, but some of them are not. And again, I specified networks. For example, you can see these rectangular, colorful things below the each panel that shows the networks. For example, the blue one is the motor control network. The red one is default mode network, and different networks. And in each network we have set of regions, and then we can see, for example, what networks are affected more than other networks. And we can go to the next slide. I think this is going to be some of the last slide, but here also another analysis that we can apply based on the graph theory to see hubs and detect hubs. By hubs, I mean those nodes that have most number of connections, and we can see if there is a change, or if any of the hubs has changed after passing some hours during the day or not. As an example, panel A is for morning, and panel B is for evening here. And there are some green circle that are depicted in the panels of the brain, as you can see. Yeah, for example, in the morning we have these hubs that are from the attention network, but they are not existed in the evening session. And in this figure, I try to show how the hubbiness and the hubs are going to be changed between morning and evening session. And I don't think we have any other slide, but can we go to the next? Oh, yeah. Yeah, I can give you a short summary. Today I talk about what is circadian rhythm, the fluctuation, the 24-hour fluctuation that existed for animal, human, plants, and most of our living organism. And the effect of circadian disruption, and how does fMRI, functional magnet resonance imaging, work. That was based on the difference of oxygenation level. If part of the brain is activated, it needs more oxygen. Then we can grab that amount of difference using fMRI. And then finally, how network science and graph theory can help us in identifying these diurnal or daily variations, just assuming between morning and evening. We can make it more complicated but because of because for this presentation and the data that we had, I just considered 2 of the sessions: the morning 1 hour after waking time, and the evening sessions 10 hours after waking time. And in the graph theory part, number 4, I try to show how we can extract the fMRI time series from these sessions and how we can construct the functional networks based on these time series. And finally, those analyses like global analysis and local analysis that we can apply based on the constructed functional networks and see how the network properties can change across the day. Yes, if you thank you for listening, and if you have any question you can ask me, or if you have any question later you can reach me out. I would be happy to help. And yeah,
Thank you very much for your presentation, and I apologize for the technical issue that we had, and thank you very much for the way that you handled it. I hope also the audience, I understand that it was not the best way possible, but I apologize for that. So we are open for any questions or any discussions.
yeah, and thank you, Dr. Tavi, for helping me to follow and moving the mouse, because yeah, I know it's a little confusing sometimes and difficult. Thank you.
Yes, sure. I'm sorry for the technical difficulty.
Yeah, no worries.
So, any questions? So I have one question. You mentioned in this last slide, in the summary slide, that we can have different sessions. Did you have different sessions, more than 2 sessions? And then if so, how the results were different, or what was the main change between those different sessions, just like those ones that you showed in morning and evening session?
Yeah, it's a good question, because we cannot have a robust conclusion just based on 2 sessions. This slide was just to show how we can apply graph theory, but yeah, it's very important to see the pattern during the day, or add more sessions, more time of day, and then we can get better and more robust conclusion. We did another analysis that we considered more than 2 times, like 6 and 8 times of day. It's like a fluctuation, but at some point and sometimes some measures are going to a peak and then decreasing, like global measures. For example, around the 4 or 5 p.m. we have some more peak in some global and local measures, and then again there is some decrease till 10 a.m. and then after that it's not easy to get the data, but it was like a pattern. There are also some papers that we published that can show these patterns as well. And yeah.
Thank you. Any other questions?
I have a quick question.
Yeah.
Were the participants engaged in any tasks during the measurements, or were they just passive?
Oh, yeah. In this study, it was just, as I said, a passive, and we say resting state. And there was no task, just resting state during the scanner, and their eyes open, not thinking of anything. And yeah, resting state. It was just for resting state.
Did you collect any data at night, or just during wake?
Yeah, not on this study, but there's another one, and the latest time that we collected the data was 10 p.m. 10 p.m. but not on this study. It's very difficult and challenging. I was not involved in any of the data collection part. I mostly focus on the data analysis, but it's very challenging to scan participants different times of day, for some even Sometimes we need to scan for more weeks, not just 1 week. And yeah.
Great, thank you. And if we don't have any other questions, I'd like to conclude this session today. And again, thank you very much, Dr. Farahani, for your presentation today. I really appreciate it. And again, I apologize for the technical issues from you, Dr. Farahani, and all the audiences. And have a good day. Thank you very much.
Thank you. Thank you.